Hello everybody, my name is Sareh. I'm doing my PhD in Department of Environmental and Biological Science at the University of Eastern Finland. Here, I would like to present a part of my PhD work which released last summer. In that work, we studied the effect of air rearing environment on sperm quality and secondary sexual traits in adult brown trout. As you know, many fish species are under threat in the world because of, for example, overexploitation or habitat degradation. And during last decades, many hatchery techniques have been perfected to produce fish more and more and release to wild. But the proportion of fish surviving to adulthood is very low. And unfortunately, endangered salmonids are not an exception. Quite often, less than 5% could survive to adulthood, mostly because of starvation and predation. So, what is the problem? Hatchery fish are different with their wild conspecifics in many important life skill traits such as anti-predator behavior, swimming ability, and migration. These are very important and affect survival in fish. And many studies have been showing that rearing method play an important role to determine growth rate and physical condition in fish. In fact, fish need to experience variation in a rearing environment to develop their adaptive behavior. Fish that adapt to new environment fast has higher survival chance. And enriched environment is one of solution. In fact, enriched environment promote post-release survival. Enriched environment is a semi-natural environment which produce developed fish with more wildlike behavior and physiology. There are many ways to enrich an environment. For example, fish are rare over natural substrates to improve their camouflage coloration, exercise fish to get ability to escape from predators or reduce rearing densities to have more natural distributions, all enhance post-release survival. It's clear now that every life conditions could affect individual phenotype. We know it as a phenotypic plasticity. Phenotypic plasticity can occur at different life stages, embryo, juvenile, and adult, and also can be seen in life skill traits such as behaviors, life history traits, and morphology. Phenotypic plasticity can also occur in transgenerational level. Um, and carryover effects can be understood as phenotypic plasticity that persists across life history stages. In fact, carryover effect is always phenotypic plasticity, always. But phenotypic plasticity doesn't necessarily mean carryover effects. Here there is an example to clarify the difference between carryover effects and phenotypic plasticity. We have two fish, they do have the same genotype, and if we put these two fish in two different environment with different amount of food, these fish feed on plenty of food and its organs are becoming larger day by day, and fish all the time needs a lot of food and behaves aggressively. Then after 3 years, we see this fish is actually much bigger than the other 3 years old, which was food restricted. Then, if we put this fish after third year to the same environment with the same amount of food, after 10 years, we can still see the differences between these two fish. It means carryover effects and phenotypic plasticity. But if after third year we put this fish into the same environment with the same amount of food, then after 10 years, we don't see any differences between fish. Weaker fish improved its phenotype. It means phenotypic plasticity, no carryover effect anymore. In our study, we predicted that early rearing conditions can impose carryover effect on adult primary and secondary sexual traits. Our study question was that can early enriched rearing produce long-term carryover effects in salmonids? This information is very important because we need to know, we need to understand how hatchery reared fish survive and perform in the wild after release. We did all of experiments in Kaino Fishery Research Station. First, we produced a full sibling offspring by artificial fertilization, and sibling groups were lately confirmed by genetic analysis. Then immediately after fertilization, we started our treatments based on natural substrate enrichment. As you see here, we had two different rearing tanks, standard rearing tanks and enriched rearing tanks, and we divided the sibling eggs into these different tanks. Then, when we had a juvenile, after the first summer, we transferred all the fish into common outdoor pool. Then we studied all male and female. Then we took pictures and measured their total length and body mass. All the fish were stripped for their gametes. We had two main analyses, sperm quality analysis and sexual ornamentation analysis. For sperm quality analysis, first we activated the sperm with water and ovarian fluid. Then we measured sperm quality parameters 10 seconds and 40 seconds after activation. For sexual ornamentation analysis, first we counted the number of spots, then we measure the skin coloration by EMPSEB program. As you see here through these two pictures, we selected two areas, one area from head and the other part of body to count a total number of skin spot, black skin spot and red skin spot. Then we selected two areas from belly to measure abdominal coloration by EMPSEB program. As a result, we didn't observe significant differences in length and weight between the treatments, and males in both treatments were heavier than females. For sperm motility, there were no significant differences in the measured sperm quality parameters between treatments, and for skin spot number and abdominal coloration, we didn't observe any interaction effect between rearing treatment and sex. The sex differed in their skin spot numbers. In both treatments, males had more spot than females. We also didn't observe differences in the number of red spots between treatments nor between sexes. And for abdominal coloration, mean hue and saturation also didn't differ between treatments. But we did find a clear treatment effect on number of black skin spot and total skin spot. As these figures showing our main results, fish were reared in standard rear tanks had more spot than their enriched siblings. And the other interesting result in both treatments, male had more spot than female. Also, we found that the color of male was more saturated than females in both treatments. Fish were reared in enriched rearing tanks had less spot than standard reared fish, and this actually demonstrated that environmental condition during early life stages can have long-lasting effect on brown trout adult phenotype. There was no difference in sperm traits, so we suggested that primary sexual traits may not be affected by early environmental conditions. In both treatment, we found that males had more spot than females, and actually, our finding supported this idea that spot likely act as a secondary sexual signal. There are many studies suggested that fish spot can be shaped by hatchery environment, and our novel finding is in line with this study to prove that in brown trout, spotting pattern can be affected by rearing environment. For example, Colorado et al. Reported that the Finnish sea trout, which mostly originated from stocking, were more spotted than the wild Estonian sea trout. As a take-home message, rearing method during early life stages affect adult skin coloration. This is most likely explained by carryover effects. And also, melanin in the skin and stress tolerance have been linked together in salmonids. As you see clearly through these two pictures, fish were reared in standard rearing tanks had more spot than their enriched sibling. I would like to thank my supervisors and everybody was involved in this experiment. A special thanks to staff of Kaino Fishery Research Station, and thank you so much for your attention.